So we only know 10% of the fungi that are out there. Only 10% of those, as just said, only less than 1% are red listed. But still, we assume that 50% are already threatened or endangered or gone. And as Anders Dahlberg, I have to steal that quote from him. It's very common in nature to be rare, and it's very rare to be common. So there's a lot of species that are very specific to their place where they live. So what are the biggest threats to mushrooms? It's us. It's habitat loss, residential and commercial development, biological resource use, and agriculture. So when we do all of those great activities, what happens is landscapes get fragmented. And you might think, well, that's not a problem. They get spores, they fly around. They will just go from forest to forest. The study that Chouette did for her master's looked exactly into that. And they found that if you go from the urban 1 kilometer out of the city, the biodiversity of spores in the air will 5 times increase, and also in the soil. So mushroom spores don't go that far, and we just heard about 20,000 sexes. So you land somewhere as a spore, and then you have to find the right partner as a spore. Is not that easy. So fragmentation is a huge problem. So it might be that this forest here is perfect for mushrooms, but they have been extinct from there for a while and have nowhere to get there. So best thing you can do for conservation of mushrooms is protect the habitat. But if you cut everything into fragmented pieces, then often reintroduction is the last resort. And as said, protection of habitat is key. And you should also not go out and just put rare and endangered mushrooms out there, because it's not uncontroversial and there's some good reasons not to do it. Imagine if somebody says, I want to protect this forest because there's this rare species, and somebody says, no, Lorin was there, inoculated, all kinds of weird stuff there. Pretty bad. So there are 10 principles, here are only 4 you should really look at before you start in reintroduction of an endangered species. Local competition. So if I bring in this mushroom species, does it take away space from others? And interaction is also not something that we very well understand. DNA verification is super important. Is this species that we put out there actually the species that we want there? Open access database in the sense of we have to know what we put where, and long-term strain preservation. It's cool if you have me, if I'm a fungi strain and I eat McDonald's for 20 years, then I don't run so good anymore. And I already don't run very fast. Yeah, so you choose some fungi for conservation. Conservation of fungi, yes or no? It's cool if you want to conserve fungi, but if there's no forest, then, and I did now with wood inhabiting fungi, so does it even make sense? And we also have to consider, is the species a good candidate for introduction? Has it been there in the past? Is it red listed? You saw the issue with that. And is it in decline? Is it actually known? And of course, strains matter in genetic diversity. You cannot just take strains from wherever in the world. You should take the strains from close by in a range of 100 to 150 kilometers, which is what they did in this project. And then also different strains, individual mushrooms can be, or fungi can be very, very different. So, yeah. This led us to 7 species of wood inhabiting fungi that are currently investigated in this project. They grow on spruce, pine, aspen, and willow. They will not try to pronounce them. But how do the fungi get into the wood? Like, how do we get them there? Well, with this help, this wonderful person, who is Chouette Crozier, who holds the mushrooms here. So, how does that work? What do they do? They looked at different methodologies, and here we have a strain growing on a spruce dowel. The first thing was, does it even work if you take a birch dowel, if you want to put it in the wood, if it originally grows on spruce? There's a lot of things that looked into. So first you have the petri dish, you have the strain that they collected, then it comes onto grain, and from there, grain is just where the mycelium then grows, and often it's dry. And then it gets either into sawdust or into dowels. And then you make a hole in the log, you want to treat, and you put the mushroom there and close it up. And so far, this has mostly been done on site. This means the researchers will go out in the forest, they find the trees, they drill a hole, close it up, they go to the next tree, which is quite labor intense. But when should you actually be inoculating on site? Well, if you have, for example, limitations on what wood can be brought into a forest, you can't just take wood from somewhere else and bring it in a very protected area because you might bring in other species of fungi. When you have the need for dead wood of high decay stages, it's really hard to drag around rotten wood. It just falls apart. Or when a mushroom grows on live trees, then you need to inoculate on site. But if you don't have to do that, you can make a mushroom farm fast. Mushroom farm, this is what they do here. And what does this mean? This means you make a cool inoculation station with tools that they build together themselves, and they collect the wood and they inoculate it on site. There's huge advantages to this. You have drill bits that are angle grinders, so you touch the wood and it goes zip, zip, zip. So you take very little time to inoculate it, which cuts the cost and the effort massively. Then they get inoculated, and for this they use special tools that are developed for mushroom inoculation. They are also full atomized. You can imagine it drills a hole, shoots in the sawdust, closes it up with styrofoam. Obviously, they didn't not use styrofoam, but they used wax. And then, after the logs have been inoculated with roughly per meter 50 inoculation points, they get brought into the forest. Sorry for the bad quality. Or in another home, where they just hang out for a while. So, what are the advantages of a mushroom farm? First of all, you can do everything in one place. That means you don't have to run through hectares and hectares of forest inoculating trees there. You can actually do it there with professional tools, and you can check the mushrooms and create the perfect environment, as you can see here with misting, for example. These are not rare or endangered species here, just as an example. And you can also observe them way better. Imagine if you have 100 trees somewhere in the forest and you have to take data on them. That's a lot of work. Here, you can all do it in one place. It's also a great way to include local communities, which is always a big thing in conservation, to include them. And not only do they look if it works, but the next step is keeping the fungi happy. So you see that it has the right moisture, it is in the right environment, it's stacked properly. And then they DNA test for colonization success. Studies like this, there has not been many, but most of them have done it in the forest, where again it's very hard to go out and collect sawdust to see if it actually grows. So you do a DNA test, does mushroom actually spreading there? And then they bring them to their final home, which means you take the logs, once they're fully colonized, they start fruiting, and you bring them out into the environment where they will sporulate and hopefully then distribute. And then you observe new fruit bodies come out, which is always a question of course, funding also. They have looked into a lot of variables of, is it dowels that work better? So these are these woody plugs with mycelium growth. Is it sawdust? Does it work better when it does this? Is it the difference what kind of wood you take? Does the mushroom care? Is it important to take fresh logs, old logs? Some of these species, Anthropocenes, I hope that is right, grows on kettle trees, which are trees, if you don't know, they grow for 200 years, the spruces, then they die for 50, 60 years, then they stand around for 50, 60 years, and they slowly just stand there, and at some point they fall over. So and that's where they grow up. So to get new substrate, that's 500 years to have new one, extinction then. Great word. So yeah, they have looked also that this species actually also grow when you put it on a freshly cut log. So what they're really striving to is enable future research into the introduction of fungi and lay the groundwork for applied projects and make it easier to include fungi in conservation projects. I think this is crucial because fungi are not considered in conservation at all. statistically not significant, one could say. No matter if it's about forestry or something else, they're just not considered. And if you want to do a new forest and you want to bring certain plants out, there's all kinds of protocols and companies and institutions you can go to who help you bring these animals back. But with fungi, there's nothing there. And it was extremely expensive so far. So I really think there's a great groundwork to make it even possible. And then also you can use fungi with the same mechanisms to use and support other conservation efforts, for example, creating habitat for vertebrates and mammals and all kinds of things. So I think it's a really great thing to do. So I have no idea what time we have, but thank you.